Hello, my name is Mohammed Hamk, PhD candidate, Sakarya University. My supervisor is Aisha Avji. My presentation is Extracellular Synthesis of Zinc Oxide Nanoparticles using Bacillus species ZPP4, and it is antimicrobial potential against foodborne bacteria. Introduction. Nanotechnology are important in our life. Nanomaterial sizes ranging from 1 to 100 nanometer at least in one dimension. Globally, the use of nanoparticles has increased in different fields from 225 to 584 tons. Unfortunately, food safety is still a big problem that facing consumers in many countries. There are three main methods of nanoparticle synthesis: physical methods, chemical methods, and biological methods. For example, bacteria, plant extracts, yeast, etc. Green technology is a promising method in the technology industry because safe, fast, cheap, doesn't need to reach high pressure and high energy, and doesn't need to additional use of chemical agents. Zinc oxide nanoparticles have been significant importance for natural sciences because they are regarded as safe according to FDA, has high capability to resistant environmental conditions, stability for long-term, daily requirement for of zinc in human 10 to 15 milligram. This study was aimed to biosynthesis zinc oxide nanoparticle using Bacillus species ZPP4 and detection the optimum condition like time, temperature, etc. Preliminary characterization of zinc oxide nanoparticles by done UV spectroscopy, and final characterization was done by FESEM, XRD, and FTIR. Finally, evaluation of their antimicrobial activity against pathogenic bacteria. Materials and method. This is a diagram that shows the activation of bacteria on nutrient agar after incubation for 24 hours. The nutrient broth was cultured, inoculate by loop and incubate at 33 centigrade with the shaking for 24 hours. After incubation, the centrifugation of with the cooling at 9000 rpm for 20 minutes. After separation of supernatant, the supernatant was mixed with the zinc sulfate heptahydrate and incubate at 33 centigrade. After that, characterization by UV spectroscopy for participate. The pills washed twice with the distilled water and followed by ethanol, and nanoparticle dried at room temperature. After that, characterization by this device. Kinetic study, we used different degree of temperature and pH, the different values of pH and different time and different precursors, concentration of precursor from 22 to 10 millimole. Characteristics of zinc oxide nanoparticles, all samples were scanned from 300 to 800 nanometer. The characterization of zinc oxide nanoparticles for morphological and size characterization, we used FESEM and EDS. After that, XRD analysis was used for detection the crystalline structure, and finally we used FTIR spectrometer for determination or detection the functional groups. Preparation of microbial suspension. 9 microorganisms were used, most of them bacteria, including gram-positive and gram-negative bacteria. Preparation of stock solution of zinc oxide nanoparticles and antimicrobial activity assay. 40 mg of dried nanoparticles dissolved in 4 ml of water, distilled water. After that, vortex for 15 minutes. After vortex, the separation of nanoparticles using ultrasonic device. After that, the evaluation of antimicrobial activity of nanoparticle using disk diffusion method. Result and discussion. The sharp peak obtained at 341 nanometer. This is figure that show the different concentration of precursor or zinc sulfate heptahydrate, and the sharp peaks were obtained at 6 and 8 and 10 millimole. This is figure that show effect zinc sulfate concentration on zinc oxide nanoparticles. The 8 millimole zinc sulfate heptahydrate was selected as best concentration for second and third days. Effect of pH on zinc oxide nanoparticles. pH 7 have sharper peaks with the maximum absorbance 3. As we see here, for all days, the pH 7 were highest than other. Effect of temperature on zinc oxide nanoparticles. Best production was achieved at 33 centigrade. This is figure that show FESEM image, and average particle size was 35.2 nanometer. This is figure that show EDX or EDS for zinc oxide nanoparticles. XRD analysis alone prod a peak at 2 theta we obtained, and which is indexed as hexagonal structure. FTIR analysis, we obtained different peaks for each of one, that mean stretching vibration or functional groups. As we see here, the antimicrobial activity of zinc oxide nanoparticle synthesis by Bacillus species ZPP4, minimum inhibition concentrations of each Listeria monocytogenes and Salmonella typhimurium were 2 mg per ml, while the minimum inhibition concentration for other species were 1 mg per ml. Conclusions concluded the biosynthesis results found that 4 factors studied like pH, temperature, etc. were significant for response variables. The optimum conditions or best conditions for synthesis zinc oxide nanoparticles were detected at pH 7, 33 centigrade, 8 millimole, and 48 hours. Also, zinc oxide nanoparticles has broad spectrum activity against both gram-positive and gram-negative bacteria. Thank you for your attention.